All right, everybody. So I know that you like the flip notes. So we're going to do one set of flip notes for this topic, and it's going to be protein synthesis. So we talked about earlier this week what proteins are and what they're made out of, but now we're going to talk about how they are actually made. So this notes packet is split into a few different sections here. So there's going to be two parts. So we're going to go through this together. So part one is the overview of protein synthesis. So our question is, how do cells know what order to put amino acids together in order to make the correct proteins? So let's go ahead and review a little bit. So DNA has the code for making proteins in its nucleotide sequence. And in eukaryotic cells, DNA is contained in the nucleus, but proteins are made at the ribosome in the cytoplasm. And we'll go over all of our cell parts in a separate video. So the question becomes, how does DNA's genetic information get from the nucleus to the cytoplasm? Through a processes called transcription and translation. All right, so the first thing that we need to go ahead and talk about is DNA versus RNA. Both of these are nucleic acids. Both DNA and RNA work together to make proteins. The DNA in the gene is the instruction for making the proteins, and the RNA uses the instructions to synthesize or make proteins at the ribosome. So the DNA is located in the nucleus. We know that DNA contains two strands. DNA contains a sugar. The sugar is called deoxyribose. And the nitrogen bases that are in DNA are adenine, thymine, cytosine, and guanine. So if we come down here and we look at our DNA, we have our double helix. Here is our deoxyribose sugar. And it's right here that the oxygen is missing on the deoxyribose sugar. RNA is in the cytoplasm and ribosome. The number of strands is 1. The sugar is ribose. And the nitrogen bases are adenine, uracil, cytosine, and guanine. So notice here that in DNA, it's thymine, but in RNA, it gets replaced with uracil. And we'll look at how that happens in a little bit here. So there are 3 types of RNA. There's messenger RNA, transfer RNA, and ribosomal RNA. So they're all abbreviated with their first letter, mRNA, tRNA, and rRNA. What does messenger RNA do? Messenger RNA carries the message of DNA out of the nucleus. I should definitely make these boxes bigger. tRNA transfers amino acids to the mRNA chain. And rRNA makes up the ribosome. And so we have an illustration of it right here. We have our messenger RNA. We have our ribosomal RNA. And we have our transfer RNA. All right, so part 1 was a little background. Part 2, diving into what is this thing, transcription and translation. So transcription is, we go DNA, transcription, mRNA, translation, protein. So transcription, the message of the DNA is changed into RNA to leave the nucleus. This is our nucleus here. This is a nuclear pore. And we have our transcription taking place where we're going from our DNA to our RNA. Notice that our RNA is single-stranded, but our DNA up here is double-stranded. So we needed to make it single-stranded so it could be nice and tiny. It could fit out of the nuclear pore. Double-stranded would be too large. So translation is the creation of the amino acid chain to make a protein. So now the next phase is this translation phase. And here are all of our amino acids connected together to make our protein. All right, so step 1, what is actually happening in transcription? To transcribe means to write. You do this in your language classes. And the enzyme RNA polymerase rewrites the code in DNA nucleotides with RNA nucleotides to build mRNA. So the substrate for RNA polymerase is the DNA chain to go ahead and rewrite it as mRNA. So what happens here is the G in nucleotides creates a C RNA nucleotide. The C in the DNA creates a G. The T creates an A, and the A creates a U, and this is this uracil. Remember, this is adenine, guanine, and cytosine. All right. So after we have done, to do transcription, we need a few things, all right? So we need the DNA template, the RNA polymerase, and the RNA nucleotides. So the DNA template is the code to make the protein. And the RNA polymerase is the enzyme that unzips and connects RNA nucleotides to DNA nucleotides. And then the RNA nucleotides that are used are adenine, uracil, cytosine, and guanine. So step 1, RNA polymerase unzips the double-stranded DNA. Step 2, RNA polymerase matches the RNA nucleotides, the DNA nucleotides on the template DNA strand, building that mRNA molecule. So if I go back and I use the colors that I had before, here's my one strand of DNA. Here's my second strand of DNA. And remember, they're complementary to each other. And then here is my mRNA that's getting made. Okay. So let's go ahead and here is our, RNA polymerase that attaches on to the specific sequence of DNA. So let's practice. The template strand of DNA is used as a guide for making RNA. And RNA uses uracil instead of thymine. So the complementary strand, so let's go ahead and review our DNA base pairing. A goes to T, T goes to A, T goes to A, C goes to G, G goes to C. You guys are really good at this, so I'm just gonna buzz along here. All right, but now we use our template DNA strand to make our mRNA. So this is where it's a little bit different. Adenine now goes to uracil. There's no thymine in mRNA. Thymine goes to adenine, adenine. Cytosine goes to guanine, guanine goes to cytosine, cytosine goes to guanine. Thymine goes to adenine, adenine goes to uracil, uracil, cytosine, adenine, cytosine, guanine, guanine, adenine, cytosine, guanine, uracil. All right, so you just made a mRNA strand. So step 2 of this whole protein synthesis is translation. And to translate means to decode. So think about any time that you're in a language class and you're going ahead and you're changing things from one language to another language, you are decoding it. So the mRNA is written in the language of nucleotides, because that's what DNA is written in, which builds RNA. And then it's translated into this language of amino acids. And you all made protein chains for the amino acids that are in your name. And amino acids build proteins. So what is needed for translation? We need a few things. We need mRNA to translate the DNA code. We need the ribosome. That is where the protein is made. We couldn't do anything without the ribosome. So if a ribosome ever gets disrupted in a cell, it's going to have a really hard time making proteins. We then have a tRNA. Okay, this is made of an anticodon to carry the amino acid. And then we have the amino acids, and we have 20 different ones. And you know that from your activity. So we have our steps here in this picture. So the first thing that happens is mRNA leaves the nucleus and it arrives at the ribosome. So this all here is our mRNA strand that has left the nucleus and it is arriving at the ribosome. I'm going to color our ribosome in pink here. Here's our ribosome. Okay. And then we have our tRNA, brings in the amino acids coded for by the mRNA. So these are all of our tRNAs. And they're all floating throughout the cytoplasm. And they're bringing all of the amino acids because the code links up. And then the amino acids are bonded together to form a protein. So these are our amino acids here, bonding together to form a protein. So the sequence of nucleotides in the mRNA determines the sequence of amino acids in the protein. That's very important. mRNA is read 3 nucleotides at a time. This is called the codon. And each nucleotide codes for an amino acid in a protein. There is the start codon, which is methionine or AUG. This is the amino acid. This is the codon. And then we have 3 different stop codons, UAA, UAG, and UGA. And they tell you when the amino acid, when the protein's ready to be stopped making. So you're probably wondering, how did I get to that? Here's our first base, our second base, and our third base. So if I looked for my start codon, it's right here. Up here are all my stop codons. All right, and then I would find the first letter, the second letter, and the third letter, and I'd know what amino acid it is. And we'll practice with that on the next page. So how are codons matched to the amino acid? So in translation, each tRNA molecule delivers a specific amino acid to the ribosome. The amino acid delivered is determined by the codon on the mRNA. The codons on the mRNA are complementary to a series of 3 nucleotides on the tRNA called the anticodon. By matching the codon on the mRNA with the anticodon on the tRNA, the cell then ensures that the right amino acid is delivered. So we're going to actually need this page to practice. So I'm going to just tuck it under the bottom here. All right, so let's practice transcription really quickly. So you translate the following pieces of the mRNA. Divide the mRNA into codons and look for each codon that represents that part of the genetic chart. Use the 3-letter abbreviation for the amino acids and separate them with a little dash. So let's first divide this up into 3. 2, 3, 1, 2, 3, 2, 3, 1, 2, 3. So if I look here, AUG. My first letter is A. My second letter is U. My third letter is G. There's M-E-T. CGU. My first letter is C. My second letter is G. My third letter is U. A-R-G. AGG. My first letter is A. My second letter is G. My third letter is G. I have another arginine. UUA. My first letter is U. My second letter is U. My third letter is A. Leucine. And then UGA, that must be one of my stop codons. UGA, here it is, stop. And you've just gone ahead and you've made a protein. So this schematic here goes ahead and shows us that here's our nucleus. Transcription is taking place in our nucleus. We have our DNA. Our DNA got opened up to go ahead and create our mRNA that's able to leave the nucleus. Our mRNA then travels to the ribosome. And then we have our tRNA molecules that all go ahead and bring the correct amino acid to each codon so that you can ultimately create a growing protein chain. And that's how a protein is made.